Hello, today I will be presenting my DNA model, which I made out of gummy bears and marshmallows, and yeah. So before I start with saying what all of that represents, I'm gonna start off with explaining DNA and what it is. So DNA is made of chemical building blocks called nucleotides, and these building blocks are made out of three parts: a phosphate group, which should be added onto here, but I made another structure showing it. So the phosphate group would be the blue ones, and that should be attached to the four bases, or to the bases that are on each line. And there's also a sugar group and one of the four types of nitrogen bases. So here's the bases, and then it just wraps around. So to form a strand of DNA, nucleotides are linked with chains with the phosphate and sugar groups alternating. So here we see that here the phosphate and then the nucleotide and the sugar group. So phosphate, sugar, and it goes in order from alternating, obviously, sorry. So the phosphate group of the nucleotide bonds covalently with the sugar molecule of the next nucleotide, and so on, form a long polymer of nucleotide monomers. So the sugar phosphate groups line up in its backbone for each single strand of DNA, and the nucleotide bases stick out from this backbone. So the atoms of the 5 carbon sugars, they go clockwise from 1 to 2 inches to 3 inches to 4 and then 5. And so as you see in this model, it shows that it goes from 3 to 5 on the right and 5 to 3 on the left. And that's what it's the backbone, it's the structure and how long it is. So the DNA is made up of 2 strands that are twisted around each other to form a right-handed helix called a double helix. So as you see, if twisted around, and if the structure would be correctly, it would keep going onward in a twisted motion. So this comes with a difference. Here comes the difference of the procreates and the eukaryotes. So procreates contain a single chromosome that is found in an area in the cytoplasm called the nucleotide. So a eukaryote contains a well-defined nucleus, but in the area, the procreates contain a single double-stranded chromosome, and then the eukaryotes contain a double-stranded linear DNA molecule packaged in 2 chromosomes. Okay, so in the prokaryotic cells, replication occurs in 2 directions at the same time and takes place in the cell cytoplasm at one point of the origin. But eukaryotic cells, on the other hand, have multiple points of origin, so they use replication with the nucleus of the cell. Okay, so also with that, prokaryotic cells possess 1 or 2 types of polymerases, whereas eukaryotes have 4 or more. So replication also happens at a faster rate for the prokaryotic cells, and replication in eukaryotics don't. It's more like less time and not that much, because obviously they have different polymerases and opposing directions and all of that. So also with that, the eukaryotics have a distinct process replicating the telomeres at the end of the chromosomes, and they have circular chromosomes, and prokaryotes have no ends to the synthesis, short replication in prokaryotes, which occurs almost continuously. So to then explain the process of replication, sorry, I have some notes here. The opening of the double helix and separation of the DNA strands, the priming of the template strand, and the assembly of a new DNA segment. So during separation, these 2 strands of the DNA double helix uncoil at a specific location at the origin. So this would be the origin. And prokaryotic DNA is arranged in a circular shape and only has a replication origin where the replication starts. But the eukaryotic DNA is linear, so when it's replicated, there's many, like 1,000 origins of replication. So RNA molecules, they are created during a process called transcription. And this process uses the enzyme RNA polymerase that goes up and down to transcribe segments of the DNA into a complementary RNA molecule. So first, the RNA polymerase binds the promoter region of the DNA to initiate the transcription. Then the mRNA, the messenger RNA, which is the one used in transcription, is transported from the nucleus to the cytoplasm. It is translated into the protein by the joint action of the transfer RNA and the ribosome, which is composed of a lot of proteins. And when it all goes in there, the transcription copies the DNA into RNA and makes another copy of DNA. So, although DNA and RNA have some chemical similarities, they all have different functions in the living organism. So for the structure of, or how transcription or translation occurs, sorry, because it's not I just exclaimed transcription, but for translation, it's that mRNA, the messenger RNA, binds into the small subunit of the ribosome, and then an initiator tRNA binds onto the mRNA. So this will be the codon AUG, which is adenine with guanine, and it just, yeah. So that's the start codon, the first codon. And then when you go to the second molecule with the anticodon, to the next codon, it will bind to the ribosome. So the ribosome then transfers, he transfers the amino acid from the first to the second tRNA, forming a dipeptide. And the ribosome then moves along the messenger RNA in a 5-3 direction. So as we see, 5-3. So this is what it's creating as it goes in the 5-3 direction. And the first tRNA is released. So another tRNA, while it has a complementary anticodon, binds on, and the chain is transferred to the next tRNA. So this process is repeated until a stop codon on the mRNA is reached and the polypeptide can be released. So yeah, that's pretty much it. So to show more of my diagram, I have, this is the red one is thymine, and then the green one is adenine, and then cytosine and guanine. And as you see, I flip these two, I flipped those, and then here I flipped these two because that's how the pattern goes, obviously. And to form a better specific, I added in the phosphate groups with the like AT, and then GC, yeah, I forgot what they're called, sorry. But I put that in there, and then I also put in the deoxyribose to help it. So I put that in there to show the rest of the structure, as you can see here. But this is my 3D structure, and I hope I kind of made sense of all of that, or in some way, but I tried. So yes, thank you. Have a nice day, night, week.